Exploration of the ensemble data. Time series characteristics of data can be obtained by fast browsing time steps. The region stability heat map view shows the stability of the selected region and provides region adjustment by directly clicking.
Deep neural networks are vulnerable to maliciously generated adversarial examples. This brings high risk in applying these networks to safety-critical applications. We developed a visual analytics approach to explain the root cause of such wrong predictions. Our contribution contains a constrained datapath extraction method, a river-based visualization, and a contribution analysis method.
In our current work, we visually compare two cohorts of segmented and classified tissue images, originated from single-cell omics data. To that end, we developed data-driven and interactive workflow implemented in a multi-linked view system. Finally, we show the effectiveness of our workflow through specific case studies.
Tableau helps you see the stories in your data. It's designed to help you be smarter, so you can make better decisions faster. Connect to the data you care about. Sort, highlight, drill down, or filter your data in seconds. Add calculations to extend your data. With Tableau, you can keep on asking questions in the data until you discover the root cause. Tableau. Answer questions at the speed of thought.
Morse complexes have shown a great utility in understanding the topology of complex scientific datasets. The noise in the scalar field, however, can significantly distort the Morse complex topology. Hence, we study the extraction of Morse complexes for ensembles of noisy scalar fields through their uncertainty visualization using our proposed statistical summary maps.
Program developers spend significant time on optimizing and tuning applications. But working with binary code to understand what compiler optimizations were applied can be challenging. We present our visual analytics system, CCNAV, designed to identify and assess compiler optimizations in binary code. Check out our paper to learn more.
KaraxS is an authoring system which enables rapid creation of zooming-based scatter plot-like visualizations in just tens of lines of JSON. We contribute a declarative grammar for such visualizations, which captures a large design space. By working with multi-node database spatial indexes, KaraxS enables interactive browsing of billions of data points. Come listen to our talk to learn more.
In this talk, we introduce CallFlow, an interactive visual analysis tool that provides a high-level overview of performance profiles from large-scale scientific applications. CallFlow visualizes the flow of resource of a program execution as a Sankey diagram to detect performance bottlenecks in parallel codes. CallFlow simplifies these calling context trees based on semantic information by constructing supergraphs.
Progressive visual analytics provides intermediate results in the middle of computation to address a long computational delay. However, such intermediate results are uncertain, so findings from these results can turn out to be incorrect for the entire data. In progressive visual analytics with safeguards, we suggest to risk the uncertainty, but we allow humans to leave safeguards every time they risk the uncertainty.
Imagine you have tons of text data to analyze. And you want to get an overview of your data, but traditional topic modeling techniques such as LDA are not working for you. Then, why don't you try Architext? We introduced a scalable and flexible way to interactively build hierarchical topics.
Digital humanities present great opportunities for testing new visualization approaches and evaluation techniques. However, and given the diffuse character and novelty of the field, it may also be intimidating for novel and senior researchers willing to get started in the discipline. In this paper, we propose a data-driven analysis of visualization for the digital humanities to identify key themes, authors, and relevant publications. So if you want to know more, please read our paper.
As data is changing, our understanding of data should be updated correspondingly. Based on machine learning approaches, we formulate a drift-level index to monitor the evolution of multi-source data, which allows users to capture and reason significant changes from time series data. The proposed visual analytics system is called Concept Explorer. More details can be found in our talk.
In our paper, we address the problem of navigating complex multiscale and dense environments, such as these molecular models. We present a technique for browsing a model by clicking on textual labels, which we call hyperlabels. This allows the user to intuitively navigate the hierarchical organization of the model. For more details, read our paper or watch the talk.
Entities and their changing relationships can be modeled more precisely as temporal hypergraphs, but hypergraph models are difficult to explore and refine. By leveraging domain-specific geometric deep learning models and a new multi-level hypergraph visualization, our technique allows for the direct integration of domain knowledge into the machine learning process. The multivariate hypergraph model structure can be analyzed on different abstraction levels. Simultaneously, experts can integrate their domain knowledge on the fly, and then explore the refined machine learning model.
Scatter plots and parallel coordinates are widely used for visualizing multivariate discrete data. However, for multivariate continuous data, the continuous scatter plots and continuous parallel coordinates are more suitable. In this work, we extend them to uncertain data. Additionally, we also extend fibers to uncertain data. If you are interested, come to our presentation.
Suppose you want to see the overall commit structure of a new project repository. Using Git class tools, GitHub Network, or Git log command is quite burdensome to get overview and navigate data. We presented Gitthrough as an interactive visual analytics system for the Git metadata to help users explore and understand the context of development history.
We present Lyra 2, a system for designing interactive visualizations by demonstration. To design interactions in Lyra 2, users directly manipulate the visualization canvas. The system interprets demonstrations using heuristics and suggests possible interaction designs. We find that Lyra 2 enables rapid prototyping of an expressive range of interaction designs. We're excited to share this work with you.
If machine learning were like education, we would like to test what concepts our student, the model, has learned. Has it learned the concept of object rotation? Does additional text help with object recognition? We need a methodology and platform for conducting such tests. In this paper, we present a novel visual analytics tool that enables hypothesis-based evaluation of machine learned models.
We demonstrate that people can use the spatial cues available in virtual reality to help them effectively remember and recall scholarly articles. We used a virtual coffee shop and asked participants to remember 4 abstracts from scientific publications, and we termed this method a virtual reality memory palace variant.
We present VIS, a tool for visualizing hypergraphs where edges can connect any number of nodes. VIS is the first technique to display hyperedges with no overlap or crossing. Vertices are represented as rows, time periods flow from left to right, and groups can be shown on the left. We designed the layout for readability. VIS shines with mid-size hypergraphs and it allows detailed analysis. We tested VIS successfully using publication datasets and data from historical documents.
We present TransVis, a design study that is proposed to analyze and integrate close and distant reading of multiple translations. TransVis presents the overview of the collection to capture global patterns that is facilitated by the ADM-VIV matrix. TransVis integrates a detailed view to explore interesting path of alignments. We also propose the TLC view to examine and explore the terms of the user selected path to justify and reason the AD analysis.
Serine graphs are variants of stack graphs with curve-based line, and the main factor affecting its readability is the sign Lorentz through a perceptual inconsistency of the orthogonal and vertical direction. Aiming at reducing its impacts, we revisited the baseline formulation and proposed the concept of composition to help the serine graph layout optimization. The result shows that our algorithms perform better than the others.
How many clusters can you see in these images? With different visual encodings in scatter plots, our perception of cluster count changes. We developed models that consider how visual density influences cluster perception. Further, we demonstrate using a threshold plot to optimize the saliency of clusters.
Taylor-Coyer flows is a turbulent fluid motion created in the gap between the two rotating cylinders. The traditional reason-based methods are only able to capture the small-scale coherent structure. The detection of the large structure depends on the 2D cross-section, which is not the true configuration. We adapt the feature-level set methods and the density estimation to combine multiple attributes and use them as the filter to separate large and small-scale structure.
We asked participants to recreate bar graphs. When the bar was alone, we saw a different pattern of error than when the bar was presented with context in a stacked bar graph. We find that absolute error increased for higher bars when they were presented alone. We also found that there was a bias in the responses such that they were repulsed from an implicit 50% mark. We found a magic number, 10%, to predict error. Participants are usually within 10% of the height of the bar.
Tableau helps you see the stories in your data. It's designed to help you be smarter, so you can make better decisions faster. Connect to the data you care about. Sort, highlight, drill down, or filter your data in seconds. Add calculations to extend your data. With Tableau, you can keep on asking questions in the data until you discover the root cause. Tableau. Answer questions at the speed of thought.
position is believed to be the most precise encoding channel, but our perception and memory of it can still be biased. Passwork has shown that the vertical position of bars in a bar chart is recalled in a biased manner, illustrating both an underestimation and overestimation bias of position. Here we find that the aspect ratios of the bar marks can cause this position bias, specifically resulting in overestimation for bars with wide ratios and underestimation for bars with tall ratios.
The classifier trained on the historical data may fail to classify the incoming data when the data distribution is changing. This phenomenon is called concept drift. To handle the concept drift, we developed Drift VIS, a visual analytics system to help the expert understand when, where, and why the drift happens, and update the model to improve performance.
The success of a convolutional neural network has been attracting many students and practitioners to learn the exciting technology. However, for beginners, the CNN model is not easy to understand. We introduced the Explainer, an interactive visualization tool to help beginners more easily learn about convolutional neural network. Using the Explainer, users can progressively explore the CNN model with real-life images in their browser, getting a comprehensive understanding of both high-level model structure and low-level underlying mathematical operations.
Out-of-distribution samples refer to the test samples not well covered by the training data, like these black cats. They are misclassified with high confidence due to their black bodies. To explain why these samples are out of distribution, we developed OOD Analyzer, a visual analytics tool which provides an ensemble detection method and a grid-based visualization to detect and analyze out-of-distribution samples.
Spot SDC is a visualization system that helps researchers understand the resiliency of HPC computation kernels to silent error corruptions. It gives users multiple perspectives of details with different granularity about the impact of SDC on an application's output. It also provides novel insight into how silent error propagates through a program's execution.
Several recent
All right, thanks everybody. This is the last session for VisInPractice. We have three great speakers. Our first speaker is Matt Harrison, who's going to be talking to us about Seaborn. Matt teaches large organizations how to leverage Python and data science at Metasync He's authored Machine Learning, Pocket Reference, the Pandas 1.x Cookbook, and the Illustrated Guide to Learning Python 3, amongst other books. He also teaches for the Stanford Continuing Studies, and he's got a great talk for us. So Matt, take it away.
My name is Matt Harrison, and I run a company called Metasnake that does Python and data science training and consulting. I've spent the last 20 years digging into Python and working with it in the data realm. So hope to have some fun with you today and learn about a tool that's pretty useful in that environment. So I'm going to be using a tool called Jupyter Notebook to just demo some of the features of this tool. If you want to follow along or you want to look at the code that I have here, it will be on the GitHub link posted here on the slides. So, brief outline, I'll give an intro to this tool, which is Seaborn, and then we'll talk about various plots that you might want to do. We'll talk about some of the features such as styling, and then we'll have a summary at the end. So Seaborn, if you're not familiar with it, according to their website, is a Python data visualization library based on Matplotlib. It provides a high-level interface for drawing attractive and informative statistical graphics. And I think it's a great library. I've used it quite a lot. I heavily used it for one of my recent books, Machine Learning Pocket Reference. I also used it in another book that I just did, Pandas 1x Cookbook. So it is super useful. Some of the features of it is it's built on top of Matplotlib and Pandas. So if you're familiar with those, you're going to get a lot of mileage out of it. It's got a lot of the common plots that you might want to use. It's got also tooling to style your plots. So, out of the box, they might not look the best. Or even Matplotlib, you can take Matplotlib or Pandas plots and style them with Seaborn. And it also has a gallery on the website. I think that's probably the most useful feature of Matplotlib and probably Seaborn as well, is that there's on both those websites, there's a gallery. You can go in there and you can check out a plot. And basically you take the code and borrow it or copy it and make your plot that looks similar to that one if you want to. Installation is pretty straightforward. If you've got pip, you say pip install Seaborn. If you have conda, you say conda install Seaborn. Okay, so let's move through what we have here. So generally, when you're using Seaborn, you'll have an import that looks something like this. In this case, we're doing this matplotlib inline, %matplotlib inline, telling Jupyter, the environment, to render the plots that we create in the notebook. So that's not Python code. That's what's called a cell magic. And then we've got some imports here. I'm importing Matplotlib because I'm actually going to use some of Matplotlib and aliasing that as PLT. We're leveraging NumPy and Pandas, and then we're going to import Seaborn as SNS. So the SNS alias there is for, I believe, West Wing. The creator of Seaborn was a fan of the West Wing show, which I've never seen, but apparently there's a person named Samuel Norman Seaborn on it, so hence the SNS. Random bits of trivia for you. And then after that, I'm just resetting the defaults. So when I reset the defaults, any plot that I make, if I've changed the defaults, this will go back to the default Seaborn behavior. Let's load the data set. I'm going to use this Ames housing data set. So there's a nice housing data set from Ames, Iowa that has various features of houses that were sold there over a period of years. And it has both numeric features and categoric features. So we're going to be leveraging this to visualize what's going on with those cells or what happened there. So just so you can see, there's various types of data in here. So we've got integers and floats, which are continuous values typically. We've got some objects. Those are string types, values as well. So they could be categorical or freeform. Most of these are categorical. So this will give us a chance to look at some different common plots that I find useful. Full disclosure, I've spent the last probably 10 years or so working a lot with structured data in Python, and I find that a lot of the data I'm working with is either of the form where it's continuous, so numbers, or it's categorical, it's strings describing something, right? I mean, there are other forms of data as well, video, audio, text, freeform text, etc. And so I'm gonna just kind of ignore those and focus on visualizing structured data. So the question I want to ask, is Seaborn a good tool for visualizing structured data, something that you get typically in a database or in a spreadsheet that we use pandas with? Okay, so let's start off by looking at categorical data. Again, this would be like, what color is your t-shirt, red, blue, or black? What size is it, small, medium, or large? Those sorts of things. I'm going to use some pandas code here to just look at the various columns in here that are categorical, or at least are string types in my pandas data set. Again, this is housing data. So you have things like neighborhood and condition of your houses. You've got, whether they had a paved driveway, what the cell condition was, what the cell type was. At the end there, you see those. Some binary numbers, whether it had like a fence or not, or a description of the fence. So these are mostly categorical values that we have in the data set, and let's see if we could visualize those using Seaborn. Now, a common visualization for categorical data is to look at the frequencies of those. So let's do that. To use Seaborn, I'm just going to say do a count plot, and here's a count plot of the neighborhood column. And to do that, I do sns.countPlot. I say along X, I want to use Seaborn, and the data that I want to use is this data frame here. Now, you see a common problem that pops up with bar plots with text labels, and that's that these are overlapping at the bottom. Another thing that we'll see here is this returns an axis subplot. So this is actually returning a matplotlib object here. So if you wanted to manipulate this with matplotlib, you can. So if you want a quick fix to get rid of that overlapping names there, is just to put it on the y-axis instead. So you can do that, just changing the x to the y there. And there you go, there's your counts of your neighborhoods from the data. So we can see that the NAIMS neighborhood has the most data. If I want to change the order of these, I can pass in this order parameter and put them in alphabet order. So you've got a whole bunch of options here. I'm not really going to dive into these, but the documentation for Seaborn is pretty good. In Jupyter, you can pull up the documentation, you can look through it. It has descriptions of all the parameters and even has examples down there at the bottom. So recommend you do that if you have questions about that. Now I'm going to show, a common option when you do a plot like that is to order those smallest to largest. And Seaborn doesn't do that out of the box, but again, you can specify that order parameter and get those in that order. We're using some pandas code to change what that order is. Now, one more thing that you might want to do, and this is a cool thing about Seaborn, is that it doesn't just have plots that return matplotlib objects, it also has plots that return these grid objects. So this plot right here, I'll put this on the next line, is a cat plot, a categorical plot. And when you plot this, note that I'm saying the column is equal to the year sold. I'm applying the counts of the neighborhoods, but I'm faceting it by column or year sold. And you can see that very easy, I can make it so it puts two columns of all the years sold, and I can compare those, make a grid like that very easily. This is not super easy to do with matplotlib or pandas. They both can do it. But with Seaborn, you get it out of the box with these plots. So this is, again, this plot is returning a facet grid instead of a matplotlib axis. Okay, so that's a brief overview of plotting categorical data. Let's look at continuous data. Again, this is numeric data. Generally, you want to look at some sort of distribution here. So let's look at the distribution of cell price. And there we go. There's the distribution of cell price. We use this KDE plot. Again, this is returning a matplotlib object. If we wanted to go in and tweak it after the fact, we can see quickly that we've got semi-skewed data here. We have like a mode around $180,000 or so, maximum up to $800,000. That's pretty good. So kernel density estimation with one line of code. There's also a dis plot, which is a distribution plot. And you can see that I'm specifying this rug equals true option here. What this is doing is putting these ticks along the bottom. Now, one thing that you might see is like the overlapping, we're running into an overlapping issue. So you might want to try and fix that. One thing you could do is just spread that out a little bit and change that aspect to spread it out. Now note that this one, again, is returning a facet grid. So it's giving us a histogram at the bottom. There's this optional rug plot that we can tack on there. And it is returning this facet grid. So that tells us we could facet it if we want to. I'll also show adjusting the ticks here. Now note that I'm adjusting the ticks. You can see that I've changed those ticks and set them at 50,000, 200,000, and 500,000. I've changed the labels there. And in order to do that, I'm basically taking advantage of the fact that under covers this is Matplotlib. So this is showing how to do that with Matplotlib. I'm changing the major locator and setting the tick labels to what I want them to be. So you can go in after the fact and change these. I can also change these from Seaborn, but I don't have such fine-grained control to change them from Seaborn. I'll show in a little bit, just changing them from Seaborn. And like I said, you can facet this. So here's an example of faceting it. I'm faceting it by year, and all I have to do is add that column is equal to year sold and the column wrap parameter there, and now I've got a faceted plot. So legitimately, this is one line of code to plot this. Could you do this using Matplotlib? Yes, you could facet it, but it would not be one line of code. It would be multiple lines of code. So really nice out-of-the-box behavior there for looking at distributions of continuous numbers. Okay, so those are one-dimensional data sets. Let's look at maybe 2 dimensions. You might want to compare 2 continuous values to one another and see how they relate. And so one of the ways to do that in Seaborn is to use this LM plot. And you can see that it gives you a scatter plot of 2 values. Here we're looking at sell price, and I'm going to shrink this down a little bit so it fits on the screen better. Giving you the sell price against the square footage in the first floor. So is this basically a proxy for how big the house is, and letting you know that it looks like there is a relationship there. It also plots a little regression line there telling you that as the sell price goes up, the square footage goes up, or vice versa. So there is a relationship there between them. Again, note that this is returning a facet grid, so if you wanted to facet this, you could. You also have what's called a rel plot, and this is very similar to the other one. It's also returning a facet grid. It's doing a scatter plot. It's not doing the regression line there. So anytime you see this facet grid here, note that you can easily facet something. And here we're going to say, okay, let's look at the price against the square footage, but then break that up into each of the years. And there we go. Now we have this broken up by years, and you can see, are there different trends in different years? And it looks like these are pretty consistent there. It's not looking like prices are increasing too much in different years. Okay, so that's continuous. Another option that you might want to compare is categorical to continuous. And Seaborn has some options there as well. So we've got one that's called the cat plot, the categorical plot. And we're going to specify what we want in the X and the Y from our data. So in this case, I'm saying, let's plot in the Y neighborhood, and in the X, we're going to do sell price from our data. Gives us this little plot here. You can pull up documentation for this, and you can see that you can pass in this kind. So there's, this is a strip plot, but you can see that there are various sorts of plots that you can do. You can do swarm plots, or box plots, violin plots, box-in plots, etc. So this is a pretty powerful plotting mechanism as well. Again, we do have that problem with the overlapping numbers down there. I'll show some tweaking of that, and we saw some tweaking of that previously. In this next example, what I want to do is do a same cat plot, but now I'm doing a violin plot instead of the default plot. And a violin plot is basically a histogram that's been tilted on its side. Well, in this case, it's not tilted on its side, but it has the top and the bottom that mirror one another. And I'm using another option from Seaborn to order to limit how many neighborhoods we're using there. So this lets us sort of dig in to a few neighborhoods. Again, this is returning a facet grid, so we can facet that if we want to. Here's an example of using a cat plot with a box plot. You might be familiar with the box plot, so we can look at the box plot to see those distributions and see how those compare. You'll note that in this example, I call XTick labels to adjust the overlapping labels at the bottom. Note that those ones are overlapping. I said use a step of 2. Basically, it skips every other tick in there and gives us that result. And again, because this is returning a facet grid, I can facet it by specifying column if I want to. So here we're breaking it out by year sold, and I'm using what's called a box-in plot, which is a fancier box plot. Basically, the tallest box in the middle is the 25th to 75th percentiles, and then each of the steps in between there is halfway between, for example, between the 75th and 100. Each step is halfway between it and 100. So it would go from 75th to 100, which would be 87.5, and then from 87.5, half that again to get all those percentages in there. So this is basically a cross between like the violin plot and a box-in plot. Pretty powerful plot. Again, basically one line of code to do this. So really featureful capabilities in Seaborn. So another thing that you might want to do is compare categories to other categories. And sadly, Seaborn doesn't really support this. So I'm going to show an example here, but this isn't really Seaborn. This is actually using pandas to do this. I'm going to compare year sold to year condition, and I'm going to use what's called a cross tabulation. And so if you're familiar with a cross tabulation, I'm basically making a little data frame in pandas that looks like this. For each neighborhood, what's the count in each of the years sold? And in this case, I'm considering years sold as a category. So there are some variables that if you look at them, depending on what light they are, they could be numeric, but they could be categorical. A year is in that gray area where it could be considered categorical. So I'm treating it categorical, but you could put any categorical data in here in this. And then basically what I'm doing is I'm doing a bar plot on top of this. So here we can see all the counts of the different cell condition types over the years sold. And if we don't want counts, if we want percentages here, this next piece is a piece of pandas code that shows me the percentages of that. And you can see the distributions that it looks like normal might be growing a little bit, whereas partial is shrinking. Now, this is using pandas, not Seaborn there. You can see that it's returning a matplotlib. Well, it's returning a legend, but if I were to take out that legend call at the end, it would be returning a matplotlib access. So I don't get faceting in this for free. I can't break this up by neighborhood or whatnot for free, and Seaborn sadly doesn't have this in it. But I do find this plot useful. I've used it quite a bit for exploring data and understanding relationships between categoricals. Okay, so that's sort of the brief gist. These are kind of the basic plots I use when I'm diving into looking at a new data set. When a client gives me a new data set, I want to sort of compare categorical data to continuous data, or look at continuous data, look at the individual columns, compare some of the columns to one another. Now, after I've looked at that, eventually I might want to give the client something, or I've even had to, create plots or charts that they will use in reports. So I want to show you some of the styling capabilities of Matplotlib that you can take advantage of. So here I'm specifying a color palette. This is just a bunch of hex RGB colors. I'm specifying a font here, and I'm specifying this style dict. And this is, Matplotlib has a bunch of configurations that you can set, and we're just overriding those with configurations that we want. And so I'm going to set my style dict here. I know that it's using a different font. It's using this Lado font here. And I'm also using a feature of Seaborn to set the context of my plot here. This basically adjusts the font size and how big things are. So there's a talk context if you want to make things that are more appropriate for talks per se. And then I'm going to change the color palette here. So I've made my color palette, and I'm going to just say use this Viridis color palette, and let's just look at a little plot. So this is a pandas plot using the Viridis color palette, and then I've got this palette that I'm printing out down here, This is also based on Viridis. So there is a color map Viridis that is built in a lot of places, but then I'm pulling out some of the points along that color map so I can plot individual lines or whatnot with that. Okay, so let me just demo adjusting our plots with some of these styling features. So if you want to change the font using Seaborn or Matplotlib, you can't use a context manager. There are context managers to let you temporarily set the style or set the color or whatnot. It turns out that you can't set fonts with context managers. Just how Matplotlib is written behind the scenes, it doesn't really like you to change the font temporarily, so that doesn't really work. So if you want to set a font, you sort of need to do it outside of a context manager. Here I am using the Lato font, and that is this font that you see up above here. It is different than the default font that we saw. So what I'm going to do is I'm going to just do my cross tabulation here. So here is a cross tabulation that's been styled. If I go up a little bit, this is what it looked like originally, which is okay. But maybe, you know, this is the company colors, this is the company font. I'm also zooming in a little bit. And so I have tweaked it so it's looking a little bit better. Okay, and here's a very similar plot. By looking at this one up here, it looks at years sold. It doesn't look like there's much of a relationship sort of pulling out there with years sold. Here, what I'm going to do is filter out by neighborhoods and look at 4 different neighborhoods and compare sales price. And here, again, we're using our Viridis color to color that, and you can dive into the code up there, but it looks like there are things sort of popping out at us, right? That the North Ames area tends to be cheaper than some of these other areas, and that you are able to see that with this visualization. So this is, I think, a pretty nice visualization comparing a few neighborhoods from Ames, Iowa. Okay, so I hope this was a good summary to some of the features that you see in Seaborn. So some of the pros that are in Seaborn is it can be great for static images because it's built on top of Matplotlib. Matplotlib basically lets you tweak anything you want to inside of it. So again, like I said, I use this in my books. I've used this in presentations. Working for clients, I've made custom themes for them. So every time I made a plot, it used their corporate font, used their corporate colors, and I didn't really have to do too much tweaking. I could just export it and they could throw it into a presentation if they needed to. It does a decent job out of the box. Like I said, some of the colors sometimes aren't really what I would call professional per se or best practices for visualization, but it does an okay job. You can tweak those. It gives you the ability to facet easily, which neither Pandas nor Matplotlib do, which can be nice. And it handles a lot of the continuous and categorical plots that I find that I need out of the box. Some of the cons, there is no interactivity here. So if you want to sort of hover over, dive in, select things, you don't really get that capability. There is some capability to do that on Matplotlib, but I don't recommend using it. I recommend looking at other tools like Bokeh or Plotly if you're interested in doing those sorts of things. Again, if you want to tweak these plots, you sort of need to have some understanding of Matplotlib. So it's not just, can I learn Seaborn and be done with it? You can if you want decent plots, but if you really want them to look good, you do need to have some understanding of Matplotlib under the covers to go in and change those. And then one thing that just sort of grates me is just the font, that you can't use a context manager. It's sort of annoying, but you can set fonts and change those if you want to. So again, I've got the link here for the code. If you want to check that out, go to that link. If you want to talk with me or interact with me, a great place to do that is on Twitter. Alternatively, I've got a link to my mailing list here. I do send out stuff on my mailing list, and also there are discounts to my courses for learning Python, visualization, SQL there as well. So I hope that you learned something about Seaborn, the Seaborn library. Hopefully you can leverage it for creating great visualizations. Have a great conference. Thanks for spending some time with me. Bye.
All right, Matt, thanks for the great talk. Seaborn seems like a really great tool. I mean, it seems like it makes the easy things easy and the hard things achievable by exposing some of the things under the covers. I mean, what really motivated the creation of Seaborn? Was it because some of the tools were too complicated to use for beginners?
Yeah, I think that's a good, interesting question. What motivated the creation of Seaborn? I mean, I'm not the creator of Seaborn, but my understanding was that basically the creator of Seaborn looked at tools like ggplot and was like, why doesn't Python have something really easy and basically one line of code to get some of these simple plots? Again, all of this is built on top of Matplotlib, and like previously, prior to Seaborn, I was faceting and making grids of histograms and plots like this without Seaborn, but Seaborn just gives you a quick and dirty interface to do that with relative ease. So I believe that was sort of the primary motivation. The other one is just a lot of these statistical plots. They, like histogram was there, but drawing a little regression line wasn't there in Matplotlib. So having a little bit more functionality, a little bit higher level interface, it seems to be nice, because a lot of people, when they're doing exploratory data analysis or something like that, really want to understand these sorts of relationships. And again, you can do it in Matplotlib. It's just a little bit more involved.
So you find it's a much more effective teaching tool because it's so easy to use, and it sort of does some of the more complicated things. Like I've done a stacked bar chart before. It's far more lines of code than it is in Seaborn than directly in Matplotlib.
So I think this gets to, like I said in the course, I don't think Seaborn is like a perfect tool. I think, and some of the comments or what I'm getting in there seem to suggest this, that it can get a little bit complicated, and that Seaborn again gives you one line of code to do a lot of things, but if you want to go in and tweak it, you do need to have some understanding of the lower level libraries. It's not just, I can't just teach you Seaborn and have you ignore Matplotlib and send you off on your way, because at some point when you want to tweak it, you're going to have to understand what's going on with Matplotlib under the covers, for better, for worse.
Yeah, so I think we've answered most of the questions that came up in chat. So there was the answer about interactivity, and it's not an interactive tool. So I just wanted to mention that, since I don't know if the comments are going to be preserved forever. But please keep asking questions in the Discord chat. And Matt, thank you very much for the talk.
Thank you. Thanks for letting me come on and share this.
All right, our next speaker is Jana Awasa, who is an assistant professor in the biochemistry department at the University of Utah. Her broad goal is to create an accurate and compelling molecular visualizations and visualization tools that support research, learning, and scientific communication. She's part of the medical animation lab. So Janet, please proceed with the talk.
My name is Jana Awasa, and I'm happy to share with you today some of my work on visualizing molecules. So when people ask me what I do, I usually say that I'm a molecular animator. And what this basically means is that I use the same type of software that people use in the film industry to animate things like fish and superheroes, except I use them to animate proteins and other molecules in order to show the kind of the hypotheses of how scientists, biologists think things work. So and other types of microscopy, like light microscopy, can tell us something about where the proteins are in cells and how they move around in live cells. And so all of these different bits of information can be used to kind of tell this story, a hypothesis of how a protein works in the cell. And so these kinds of stories are typically drawn out in what's called the model figure. And these are some examples of model figures that were drawn for me by collaborators. So I first got interested in molecular animation as a graduate student. I was in a lab that studied cell motility, how cells crawl, and we were right next to a lab that studied these proteins known as kinesin that are known to walk along the kind of a highway in the cell, microtubules, and carry things from one side of the cell to the other. And so we had joint group meetings, which meant that every week I would watch a presentation by a graduate student or a postdoc about how kinesin worked. And typically, these presentations included these types of figures, like the ones I'm showing here. And most often, these figures show some kind of a very simplified depiction of a molecule. In this case, kinesin is shown with two triangular kind of heads that basically are kind of touching a microtubule, which is shown by that line. And so this is kind of showing how a microtubule, so how a kinesin walks along a microtubule. And from watching lots of presentations and seeing a lot of figures like this, I felt like I really understood how kinesin works. But then, you know, during my grad school career, a couple years in, the structure of kinesin was solved by this lab, by the Vail lab and the Milligan lab. And they decided to hire an animator named Graham Johnson to create this visualization of how they thought kinesin walked. And the first time I saw this which was also during group meetings, so kind of in the context of a kind of a scientific presentation by a graduate student, I was kind of blown away. I was blown away by the information that it contained, and also by the fact that it was just so intuitive. I think I never really totally understood how kinesin moved until I saw it in this sort of form. And so it really made me start to think about why we weren't creating more visualizations like this, why we were, you know, relying so heavily on these types of simplifications when our data was really pointing us more to these types of visualizations that can incorporate dynamics as well as a three-dimensionality, which is kind of what our data was really describing. So by the end of grad school, I was really convinced for the need of these visualizations in cell biology, in molecular biology. And, you know, I was really in love with the idea of using 3D animation to synthesize diverse biological data. And that includes data on protein structure and activity and dynamics, all of these different things that we're collecting data on, and enabling us to really create an idea of how things work. And then the animation could really be this sort of visual hypothesis of how we thought or how we think these processes occur. So I've been working on this type of work for well over a decade now, and I've had the good fortune of working on a lot of really interesting projects. That includes animating cell motility from my grad school years to more recently working on things like CRISPR and how gene engineering works. One of the kind of most long-running projects that in my lab right now is a project known as the Science of HIV. This is kind of mainly an outreach project where we're thinking about how to convey complex information to the public. And so this is really a collaboration with a P50 center, an NIH-funded center that's housed at the University of Utah. And I have collaborators within this group in a lot of other places in the US. And the focus is really on the structural biology of HIV. So how is HIV able to get in cells? How does it get out of cells? And what are the kind of molecular machines involved in that process? And so this animation, so I've created an animation of the life cycle of HIV, which is constantly getting updated as we have new data. So I'll show you a bit of this animation. So the animation starts by showing the virus and what it looks like. So this is kind of the basic anatomy of the virus. You have this sort of membrane, which is shown kind of in this light greenish color, and you have these envelope proteins on the outside of it. And inside of the virus, we have this conical shaped capsid, which houses the RNA, which is shown in blue. So this is the genome of the virus. And one of the things that I wanted to show is the relative scale of the virus next to the host cell, which is a T cell in this case, to show how much smaller really the virus is. The first part of the life cycle is entry. So how does the virus gain entry into the cell? And this is carried out by basically proteins on the viral surface known as envelope proteins, shown in blue and red, which are able to make contact and bind to these proteins on the surface of the T cell. So these proteins are shown in yellow and orange. And when these proteins all make contact with each other, the virus protein, the envelope protein, is able to undergo a series of dramatic shape changes, which then basically causes the fusion of the membrane, the viral membrane, with the cellular membrane. And this allows the capsid to gain entry into the cell. And so this animation basically walks through all of these different parts of the life cycle at a molecular scale. So just kind of briefly, we have, while the virus, the capsid is moving towards a nucleus, it's thought that you start to copy the RNA genome into DNA. So you have double-stranded DNA, a double-stranded DNA copy of the viral genome. This gets integrated into the nuclear genome, so basically the genome of the cell. And that enables the regular cellular machinery to start turning on these genes, these viral genes. And so that process is shown here. This DNA is transcribed into RNA, which then gets transported out of the nucleus and then can be used to make proteins, and also can be used to get packaged into new viruses. So at this point, the cell is considered to be sort of a viral factory, and it's now creating new viruses that can now bud from the cell. And so I'm gonna slow down a little bit here and focus on these little teal proteins that are shown at the neck of the virus, this new budding virus. These proteins are known to be involved in fission, so basically cutting off the virus from the cell and releasing it into the extracellular environment. And the last stage is known as maturation. So initially when the viruses are released, they don't really look like the mature viruses that we saw in the beginning of the movie. But however, soon after they're released, they undergo a series of changes, shape changes that then allow this capsid to form. So this animation is available, publicly available on scienceofhiv.org to download. So if you're interested, you can watch that at your own pace. So one of the things I've been interested in within the context of this HIV project, as well as many others, is thinking about how we can incorporate animations into research. One of the things that has been really kind of an interesting thing to see is that animations can really highlight how different hypotheses differ. So if we focus just on SCORT3, those proteins that were involved in the fission step of the virus, the viral release, there are actually different hypotheses of how this works. And so this is kind of nicely animated or illustrated through animation, where in this case we have we can show three different hypotheses that different researchers have come up with about how this fission process works. And so that's one, I think, really useful way of how animations can be really helpful in research. And this was published in a review paper with a collaborator, Jim Hurley at UC Berkeley. Another example, again, on this escort complex, which is a pretty hot topic right now. in these fields is this paper. So this was a recently published paper from another collaborator, Adam Frost, and a postdoc in his lab, Henry Wen. And they were studying these sorts of proteins that can, the escort proteins which can undergo, they saw these conformational changes. And this is the movie that they created from their data. And one of the great things about animation, so I created this animation for them to basically contextualize the structure. So the structure they have is basically two snapshots, one kind of a more open state, and one is a more closed state. But the hypothesis is that there's the transition between these two is initiated by binding of another protein that then kind of forces this change to go from this more open to a more closed state. And so that is kind of shown in this animation. So the animation can really provide a little bit more context and the dynamics to show kind of fill in the blanks of a hypothesis. So what is the process of these animations? So in a lot of ways, it's very similar to what you might see at an animation studio. So the first step is typically storyboarding. So actually the very first step is just having a conversation with a research collaborator about what the process is that they'd like to visualize. And then we go through this stage of storyboarding. In this case, this is my PhD advisor who created this storyboard. It's a really nice storyboard. And it kind of shows step by step what should be happening to the molecules at each stage. Typically after that has gone through kind of a back and forth process, we typically draw storyboards and use that as a means to kind of make sure that we have all of the details right with our collaborators. Then we go into modeling. A lot of the times we have at least partial structures for a lot of the molecules that we're animating. And so in this case, we're using a software known as UCSF Chimera in order to create these sorts of meshes over the proteins. So the proteins themselves are usually in the form of a protein data bank file, which is a series of coordinates, XYZ coordinates for each of the atoms in a structure. And we then use that to create these meshes that then could be imported into our software. Chimera can also be used to create meshes around light microscopy images. So in this case, this is a cell that has been imaged using light microscopy. And then a mesh has been created in Chimera and then kind of integrated into this animation to provide context to what molecular events are happening within it. So during the animation stage, there are a lot of times when, we really have to kind of be sort of inventive in thinking about ways we can use the tools that are there to animate things at a molecular scale. So this is an example where I'm using hair to model RNA and to animate it. So this was used in the HIV animation, where basically you have the RNA that's the virus that's undergoing maturation, and the RNA is thought to kind of glom into the middle of the maturing virus as it gets encapsulated in the capsid. And so the hair using different forces on the hair allowed me to kind of create that effect that would look relatively convincing in how researchers expect it might look. All right, and so yeah, so this was that animation. You can see kind of that RNA in the middle there before the capsid comes around it. So the last part of the talk I wanted to talk about was basically kind of the issues with animations and the ways that we're thinking about trying to address them. So one of the major issues that researchers have pointed out and really kind of gives a lot of people pause within the molecular biology community is that, animations, it's not, you can't really tell from watching an animation what things are really well understood or that we have a lot of data to back, and what parts of the animations are really kind of more conjectural and guesswork. And I think that, the truth is for any molecular animation, there will always be a mix of things where some structures, some events, some processes may be very well supported by lots of different modes of experimental data, and other parts are really not. And the question is really, how do you show that within the context of an animation, and especially, are there any good ways to do that right now? So that's one of the things that has been kind of something that I've thought quite a lot about. And so, we've been able to address this issue to some degree very recently through a recently funded NSF project on coronavirus. And so this project is similar in many ways to the HIV lifecycle animation, except now we're trying to animate coronavirus. And coronavirus is interesting for a number of reasons, but one of the things that has been really remarkable about coronavirus is the rate at which people in the biology community are releasing data on coronavirus, the structures, the life cycle. And so we're really learning a lot about coronavirus quite quickly. And so the goal of this project is twofold. The first part is really to create this animation of the life cycle, and I'm gonna just play through this very early first draft of entry. So this is, again, is kind of like an anatomy visualization of the coronavirus, showing kind of all the different parts, including the RNA genome, and all the envelope, the kind of what's called the spike proteins on the surface. And so the animation walks through the surface. So this is the surface of a respiratory cell, and has different types of proteins on them. Some of these proteins are proteases, and some of them are the receptors. So the purple proteins are the receptors. They're called ACE2. And these yellow and orange proteins are proteases, which are thought to be necessary in order for the spike protein to undergo these conformational changes that lead to membrane fusion, as we saw before. And so that's what's shown here is a series of these sorts of cleavage events that then lead to the spike protein undergoing some pretty dramatic shape changes that are shown here that are kind of similar in many ways to what we showed with HIV, which then leads to membrane fusion. So this is just the first part of the life cycle of coronavirus, and we are in the process of animating the other parts, what happens inside of the cell. And this is interesting to work on because it's, again, a lot of moving parts with this animation, and a lot of different collaborators around the world, many of whom are relatively new to the coronavirus field. So the second major part of this project is thinking about annotation and commenting. So what we're really envisioning, so this part of the project is being carried out by Jen Rogers and Mariah Meyer and Alex Lex's lab at the University of Utah. And what we're really thinking about is how do we annotate this animation to make it clear where we got the data to create these models and to create the types of movements that we show. So how do we annotate that in terms of papers or structures or even just conversations we've had. The second part is, so here on this screen that is a kind of a prototype of this software where the left side shows this the annotation and the right side is the comment. So in this first phase, we basically showed this animation to a series of experts on the coronavirus entry, and we asked them to comment on whether they thought this was looking correct, whether this was looking incorrect, what they would change, and we got a lot of advice on how this could be changed, especially with new data. And so I'm currently in the process of creating a second draft of this. And I think so the commenting tool is really kind of the most interesting part in many ways. It enables the scientific community to really discuss this. And the idea is to think about how we can move towards a consensus model of the coronavirus life cycle. When do we know when we have more of a consensus versus a lot of disagreement where there's a lot of unknowns? So this is one project that I'm really excited about. Within the next 6 months or so, we'll release different parts of the life cycle, and so it's definitely something that if you're interested in this, you can keep an eye out for that. So that's the end of my talk. Hopefully that was interesting, and feel free to get in touch with questions. And this is kind of my thank you slide. I'm gonna move this so you can see everybody's names here. I'd like to thank members of my lab, people who have collaborated with me on different projects, and for funding. Thank you.
All right, thanks, Janet. That was a great talk. There was some really cool visualizations. I'm very impressed.
Thanks.
So we have some questions coming in from Discord. So one person is asking, could these animations become short research films, and are there other researchers comfortable with recording voiceovers or writing documentary style captions for these?
So nearly all of our animations are essentially research movies. So they're kind of the hypothesis of our collaborators. So they are used in publications, in research talks, and yeah, they do sometimes. The researchers in general are really shy about providing voiceovers. I don't know why, but I often have to record them myself, or have someone in my lab record them. So they're happy to write narration and stuff, and the ideas are entirely theirs. Exactly what you see is kind of what they hypothesized. So I think it's kind of more, my goal is to try to make this more commonly a way that people use visualizations to communicate their ideas in biology.
So it seems like there's significant investment when you decide to make an animation. So like, what is that cost? And I mean, what tools are you using, right? So on one side, you have like something like Houdini, where it's fully featured, made for the rendering and animation industry, but then you have all this data wrangling that you have to do from the data that you guys have. I mean, how do you bridge that, and how expensive is it?
So the software itself, we typically use Maya for the animation. And so we use an educational version. We make all of our animations are essentially free for the research community. A lot of them are publicly distributed. So I think in terms of cost, it's people time, and that can be expensive, of course. So what we're looking at is an average animation takes a postdoc of mine in my lab, maybe months to complete. And so we're really kind of looking at their time to complete that work within that time period. So there's not really a ton, I guess, of kind of like, cost in terms of the software. I guess there is a cost in terms of the hardware, but it really is mostly effort. So most of our grants and our funding funds the effort of myself and the people in my lab. Does that answer your question, or not quite?
It does answer my question. I was mostly, I wasn't talking about the cost of software. I was really asking about the effort involved, because it seems
so it's
very
usually
time consuming.
months. Yeah. So I think a lot of the times, like we generally don't just select projects. We usually have collaborators come to us with a specific need. Usually they've been, trying to work on communicating an idea for a long time and haven't been successful. So for them, finding that funding and being able to fund that kind of effort is worth it.
So we have another question on Discord. I'll distill the question a little bit. So basically the question is asking, have you considered alternatives from existing tools for annotation software similar to some things that have already been published?
Do they have examples, or I guess I'm not sure.
They do. So software proposed by Jenkinson and McGill.
Right. So they have proposed software, but as far as I know, there is no software that actually exists. So we've actually talked to them. We had a workshop recently where we brought in a bunch of different medical illustrators and animators, including Jody and Gail, to talk about the annotation and what ideally we would have in an annotation software. So this was a workshop that Jen Rogers and organized on in Mariah Meyer's group. And so I think, yeah, so we're hoping to have something that the community can use, but there actually isn't really an annotation software out there. I think there are ideas of the fact that we need annotation software, but the actual software doesn't exist.
So in your talk, you mentioned the ability to describe what is certain and what is uncertain. So essentially uncertainty visualization for the animations, like what is well understood and what is not well understood. I mean, do you address that through the narration of the video? I mean, how do you approach things like that?
So I think, that's one of the reasons why, of course, we need an annotation software or commenting software to be able to point out where we get our data for the animations, because we aren't able to easily communicate that, whether there's a consensus model that, a majority of the community might agree with, or whether there's really just kind of one rogue idea out there about how things work. It's really difficult to convey that through an animation, which is why I think this sort of annotation or commenting software is really necessary. But I think prior to having that, I really rely on my collaborators. So most of our work is, kind of meant to be embedded within a research talk. And so you can usually tell from within, it's at the end of a research talk, a biologist is talking about their data, they're showing a bunch of images, they're showing a bunch of things. And at the end they say, and this is our model of how things work. And I think if you're watching the rest of the talk, where the data actually came from and what's actually a little bit more conjectural. So that's usually what we rely on, is for our collaborators to make it clear what actually came directly from their own data, and what may have been kind of more connecting the dots. But I think, it's not always great because a lot of the animations can be taken out of context. They aren't always presented in that sort of a research presentation like that. And in that case, the animation gets totally disconnected from the data it was derived from.
All right, excellent. I really enjoyed the talk. Thanks, Janet, for coming to VisInPractice.
Sure.
Our next speaker is Dr. Steve Maas, who is a research associate at the University of Utah. He has developed, along with Jeff Weiss of the University of Utah and several other developers, the FEBio software, which is a finite element package for applications in biomechanics and biophysics. He has also developed FEBio's supporting software packages, which includes supporting VIS. So without further ado, Steve?
Hello, everyone. My name is Steve Maas, and I'm a research associate here at the University of Utah. I am one of the software developers of the FEBio project, which I would like to talk to you about today. More specifically, I will be talking about visualization of data and results for the FEBio software. So FEBio is a finite element solver that was specifically designed for solving problems in biomechanics and biophysics. The development of FEBio is a collaborative effort between the University of Utah, where it is led by Dr. Weiss, and Columbia University, where the PI is Dr. Ateshian. The project started around 2005 when we and other researchers recognized that there was a need for a finite element software that was tailored specifically to the needs of the biomechanics community. At that time, most researchers were either using commercial packages, which were designed for more traditional engineering applications and were often limited in their capacity to model problems in computational biomechanics. This led some researchers to develop their own finite element code, which was obviously a very labor and time-consuming effort. Many of these codes were not general purpose and often didn't make it into the public domain. So with FEBio, we kind of wanted to fill that gap by developing an FE software package that, on the one hand, offers ready-to-use FE software rich in features relevant to the biomechanics community, but on the other hand, since we recognized that computational biomechanics was, and still is, a rapidly evolving field, we wanted the code to be relatively easy to customize. The first release of FEBio was done at the end of December in 2007, and we currently have over 12,000 users and have over 60,000 downloads of the FEBio software itself, which does not include all the supporting software that we developed. And we also try to keep track of publications that have used FEBio and have counted so far over 500 publications. To give you a brief overview of the key features, with FEBio, users can model structures ranging across different physical scales, ranging from the cellular level to whole body mechanics. FEBio is a true multi-physics code where mechanics, fluids, mixtures, chemical reactions, and several other types of physics can be combined in a single model. We offer a large variety of constitutive models that can be used to model the often very complex material behavior of biological tissues. Users can also include dynamic effects, residual strains, active contraction, fluid and mass transport across interfaces, contact, and much more in their models. FEBio is used in many research fields, including soft tissue mechanics, joint and muscle mechanics, cardiovascular mechanics, and eugenesis research, just to name a few. Of course, we use FEBio ourselves in several of our research projects. As I mentioned, FEBio is a collaboration between the University of Utah and Columbia University. At Utah, we use FEBio for studying hip joint mechanics and eugenesis, which is the formation of new blood vessels and how it affects the mechanics of the tissue. And we're also working on the characterization and visualization of complex fiber distributions. On the Columbia side, Dr. Ateshian works mostly on cartilage mechanics. He has also done a lot of fundamental theoretical work and has contributed to many original formulations in areas like damage mechanics, growth and remodeling, but also fluid mechanics and FSI, and all these formulations are implemented in FEBio. So just to give you one example of how we use FEBio in our research, I just want to present this work. So this work is part of one of Dr. Weiss's PhD students, who is looking at the characterization of cartilage mechanics in hip joints. She's trying to understand how different loading conditions affect the stress and fluid pressure distributions in the cartilage. Peak values are often indicators of pain, and so this research can help surgeons develop a better understanding of the causes of these pains and make better decisions in when and how to treat patients who suffer from diseases such as hip impingement. Coming back to FEBio, it is important to understand that FEBio is a command line solver and uses a file-based communication mechanism. In order to make FEBio easier to use, we also developed our own preprocessor for generating the FEBio input file and postprocessor for looking at the simulation results. Originally, these were two separate packages, but recently we integrated them both into a single FE modeling software, which is called FEBio Studio. A typical workflow in FEBio Studio goes something like this. So first, users import their surface or volume meshes. In the case of surface meshes, they can then generate volumetric meshes. They can also check mesh quality metrics and repair some common issues. Next, users define the physics of the model by assigning different materials to different parts, setting up boundary conditions, contact conditions, rigid body kinematics, and so forth. Then FEBio Studio will generate the FEBio input file and we'll call FEBio, and users can monitor the progress in FEBio Studio. And then finally, the results are loaded back into FEBio Studio for visualization and animation, analysis, and data extraction. So what I'd like to do in the remaining of the presentation is basically illustrate how that we use visualization to help the user through the different parts of this typical workflow. So starting with the mesh definition, FEBio supports many different types of elements, including solid elements such as tetrahedral, hexahedral. It also supports shell elements such as triangle and quadrilaterals, and many of these elements can be linear, quadratic, or even cubic. Meshes can have different element types, so they can combine all these different kinds of elements. And so, of course, we need to have algorithms in FEBio Studio for rendering all these different types of meshes. Mesh resolution and mesh quality are very important in finite element modeling, since they directly affect the accuracy of the solution. In large deformations analysis, particular care must be taken to produce good meshes that do not create element inversions. And invert elements are basically the elements that are turned inside out, and when this happens, FEBio and other finite element software, for that matter, will not be able to continue. So it's important that users can inspect the mesh quality a priori, and FEBio Studio offers several quality measures that they can check. Color plots are typically used to visualize these different quality measures. It sometimes happens that imported meshes contain problems that will prevent them from running correctly, or even from generating a volumetric mesh. So typical issues are small holes, invalid triangle topologies, elements that have incorrect node numbering, since not all meshing software uses the same conventions. So these issues are not always easy to spot, so we try to offer visual clues by generating outlines that define what we call crease edges. The crease edge is basically drawn anywhere where the software detects some kind of discontinuity or sharp transition. And then combined with transparency effects, this can often be very useful to quickly identify any issues with the mesh. Orientation of surface normals can also be important for mesh generation and for certain types of analysis like contact problems and surface loadings. So to visualize the normals, FEBio Studio draws short colored line segments on each facet that are RGB color coded, by which I mean that the color is determined by the XYZ coordinates of the normal. So the X component defines the red color, the Y component the green color, and the Z component the blue color. So for example, a red vector would point in the direction of the X-axis. On the model definition side, we use visualization to help users ensure that the correct physics was assigned to the correct part of the model. For instance, color coding is used to identify which materials were assigned to different parts. Stencil-based transparency is used for selecting and highlighting. Structural mechanics models often contain discrete components in addition to deformal components, and by discrete components I mean springs or dashpots, or rigid bodies that are connected via joints or rigid connectors, and so forth. And in FEBio Studio, we use glyphs that are drawn on top of the model to indicate the type, the position, and the orientation of these discrete components. Many constitutive models used to represent biological tissues are anisotropic. Anisotropy is often induced by one or multiple embedded fiber distributions. These distributions are often generated procedurally, so it's important for users to see how the algorithm oriented the fiber in each element. And to visualize the fibers, we use simple line renderings where the line segments are colored using the same RGB color coding scheme that I mentioned before. Once the model definition is done, FEBio Studio sends the model to FEBio. At this point, there's not much visualization going on, of course, but the only thing I want to mention here is that users do have the ability to load partial results into FEBio Studio, and this can be helpful for catching errors in the model definition early without having to wait until the entire simulation ends. That brings us to the post-processing side, where most of our visualization algorithms can be found. The solution of most FEBio models will have multiple time steps, even for problems that are not time-dependent, and this is a consequence of the fact that when dealing with large deformations, it is often easier to find the solution if the loads are ramped up gradually. As a result, users need to be able to step through or animate the different time steps of their model. The most commonly used visualization method in FEBio Studio is color plotting, where users can create color plots of some physical quantity of interest, such as displacement, strain, or stress, in the case of a structural mechanics problem. Since the area of interest may not lie on the outside of the mesh, users have several options for looking inside the mesh, such as hiding parts or elements, using plane cuts, or transparency. FEBio Studio also offers a screen capturing feature. This is necessary since for large models it becomes impossible to render at high frame rates, so instead users can capture the individual frames of the different time steps and then save the resulting images to a video file format. All the videos in this presentation were created in this manner. I mentioned earlier the importance of being able to visualize fibers on the pre-processing side, since due to large deformations, the fiber distributions themselves may have changed quite substantially. It may be of interest to visualize these fiber distributions on the post-processing side as well. Since fibers are defined by vectors, users can use various types of vector plots to visualize them. The vector glyphs can be colored by some scalar measure, such as fiber length or orientation. Users have the ability to overlay line or particle data that was obtained elsewhere and added to their FE model. So this is actually something we do quite often in our angiogenesis visualizations, since the vessel networks, although obtained during the FE solution, are stored and imported separately from the FE mesh data. So in this example, we see tiny vessel sprouts growing inside a constrained region and inducing stresses and deformations in the surrounding tissues. I already mentioned that FEBio can do fluid and FSI simulations, so we implemented various visualization methods for displaying fluid flows. These include contour plots, here used in combination with a plane cut to show the fluid velocity in the interior of a bifurcated artery. Streamlines can also be used to visualize fluid flows. We use automatic seeding methods to make it easy to generate the streamlines. Users can also create particle flow plots, as can be seen here. Again, automatic seeding is used to start the particle flows. And we also have algorithms for rendering the volumetric flows. So all these different visualization methods can be combined in a single visualization. In this example here, the color plot is used to indicate the fluid pressure in this pulsed flow example, which is an FSI example that shows that the flow is producing deformations in the surrounding tissue. And the vectors here represent the fluid velocity. So this brings us to our current work, at least as far as visualization in FEBio Studio goes. The main focus lies currently with representing heterogeneous model parameters such as material parameters, which essentially means making the parameters dependent on the position. A big challenge here is how to generate this data in the first place, and FEBio Studio currently offers the option to generate the data via a mathematical expression or using the solution of a Laplace solver. So this basically means solve an FE problem on the mesh, which produces a scalar potential field. This field can then be assigned to a model parameter. Vector fields for defining fiber distributions, for instance, can be created by then taking the gradient of this scalar field. We also want to add the ability to extract model parameters directly from image data. Obviously, it will be important to have clear visualization to represent all these different fields for these heterogeneous parameters. Now, fortunately, we already have a lot of visualization algorithms on the post-processing side, which we can port to the pre-processing side and where they will be used for this purpose. Now, I want to say a little bit more about the option to generate data from volumetric images. So we're currently working on integrating image data into the entire simulation pipeline. Usually, image data is only used to create the surface meshes via image segmentation, but image data can have value in other parts of the pipeline as well. As I mentioned, it can be used to define heterogeneous material parameters. For instance, it's well known that CT image intensity correlates to some physical properties such as bone density or stiffness. And currently, users such as this user here, who's working on this femur model, would have to generate this data elsewhere and then bring it into FEBio Studio, which is possible, but it often requires some scripting to handle the necessary file conversions. So if we can bring the image data into FEBio Studio, we could do the mapping ourselves and save users potentially a lot of time. So although we're still working on this, we have already implemented several visualization methods for displaying volumetric image data, including showing image slices, doing volume rendering. This can be seen here in this video, and also isosurface extraction. The volume image renderings can be combined with the image renderings, as can be seen here in the image on the right, where volumetric image data of the heart is rendered, and several arteries represented by surface meshes are overlaid on top of the image data. So this last example was used in a study that uses hyperelastic warping to create patient-specific heart models. So hyperelastic warping is a finite element-based deformable image registration method, which was also developed here in Utah by Dr. Awasa and his colleagues prior to the FEBio development. Hyperelastic warping is currently implemented as a plugin to FEBio. And the reason I wanted to show this example is because it's a great illustration of how image data can be used in the entire modeling pipeline. So in this study, XCAD phantom CT images were generated from the living heart model, which is a fully functioning model of the heart developed by Dassault Systèmes. And in this particular work, only the geometry of the heart model was used. Then patient-specific CT data was collected and then modified to match the XCAD image intensities. Hyperelastic warping was then used to deform the living heart model onto the target's patient-specific image data. So that brings us to the end of this presentation. Now, before I wrap up, I would like to acknowledge my fellow team members, including the project PIs, Dr. Weiss and Dr. Ateshian, as well as my colleagues, Dr. Ben Ellis and Michael Heron. I also want to give a big thank you to all those who have contributed models, images, and videos for this presentation. And I want to thank our funding sources for giving us the opportunity to work on this exciting software project. And then finally, I want to thank everyone in the audience for watching my presentation. If you would like to know more about FEBio, please visit us at www.febio.org. You can find all our source codes on GitHub. If you would like to stay up to date on FEBio related news, make sure to subscribe to our Twitter feeds and our YouTube channel. Thank you.
All right, Steve, thanks for the great talk. That's a very interesting software package. I have some questions for you.
Okay.
So at the beginning of the talk, you mentioned that these were originally two distinct pieces of software, one for the simulation and one for the viz. What was the motivation for combining them into a single software package?
Right, I think that the main motivation was just making the software easier to use. So before we had an integrated software package, that users had to basically learn 3 different software packages. They had to learn the preprocessor, they had to learn the solver, which was the command line application. So they have to figure out how to run command line applications on a computer, and there was some challenges there in terms of setting up environment variables and things like that, and then they had to learn the post-processing side. And so after many years of doing it that way, we kind of realized that this was not the most ideal way for people to get started with the software. And so for that reason, we decided to combine everything into a single software package called the FEBio Studio. Now, people who were using the old way of using, the GUI for creating the models, but the command line for solving the models, they can still do that. In some situations, that's the only way to do things. Like if, for example, on Linux clusters, you still need to use the command line interface. But we think that the new approach of putting everything in a single application is going to be a lot easier for new users to get started with FEBio Studio.
So I had some more questions. So one of the things you pointed out was that the element inversions are a significant hindrance into the overall workflow. Like the elements can tangle and then they have to go figure it out. So I mean, you mentioned that you can also look at intermediate results. I mean, how is that? What does that overall workflow look like from running sim, checking intermediate results? What are they looking at? Are they looking at the same mesh quality metrics that they were looking at the beginning when they're creating the model? And then how do they go back and fix it?
Yeah, so that's a good question. And so when you're monitoring, you're just basically seeing if everything is going well. So you're not really monitoring mesh quality metrics as the simulation is running. You're just monitoring to see if anything is going wrong. Like if something goes wrong, like an element inversion, the software will tell you that. Even if you're not monitoring, you will still get error messages. And we have some methods that we try to recover if in case that happens. So we can try things like reducing the time step sizes and then just try it again. Sometimes that doesn't help and users have to go back and change some model parameters in order to try to get around problematic areas in the simulations. And in worst cases, they have to go back to their mesh generation software and try to find a mesh quality or a mesh density that works better for the kind of simulations they are running. So there's not really a foolproof answer to this, or a strategy that will guarantee success. It's often a bit of trial and error, and then with experience, learning, try to anticipate these problems so that you can avoid the most common challenges in these kinds of simulations.
So going back to the different components of FEBio, I mean, so what is the effort investment in terms of the mesh generation, simulation, and the VIS part? So like, are they sort of spread out equally, or does one dominate the others?
I think mesh generation these days is challenging. It's still challenging, but perhaps not as challenging as it used to be. So there's a lot of semi-automatic segmentation that people use these days to generate the surface meshes. It's relatively easy to generate the volumetric meshes, and then it really depends on the kind of simulation that you are running, whether or not the actual simulation is going to be time consuming. So if you're doing structural analysis kinds of problems, then it really depends on how well conditioned your simulation is. There's other challenges involved when dealing with simulations in fluid mechanics and FSI and things like that. But I'd say that I would say maybe half of the time you spent setting up your model, including mesh generation, and the other half of the model you're trying to run it and fine-tuning parameters.
And what about from the development side? Like, how much effort have you poured into each one of these tools?
To be honest with you, I'm not sure. I haven't kept track of how much time I've spent on different parts, so it's difficult to say. I think the most challenging part is definitely the FEBio site with all the physics stuff. But I think a lot of that physics is now being done by my colleague Jana Awasa, and most of the work that I spent now are on FEBio Studio. So I'd spent maybe 80% of my time on FEBio Studio and 20% on FEBio, I would say.
Cool. So I guess the last question I have for you is, so you, in the talk, you mentioned that you support not only linear elements, but quadratic and cubic elements. So when you do the VIS, are you rendering those higher order elements natively, or are you how are you dealing with them?
So basically, the algorithm kind of creates a very simple linear triangular mesh of each facet of the higher order element. And so it renders that triangle mesh for each element. And, for a big model, this can be, of course, very problematic in terms of the number of actual triangles being rendered. So there's a few settings that users can control in terms of how accurate they want to see these higher order elements being displayed. So, and there's also some automatic checking. If the number of elements is fairly low, then we try to render it as accurately as we can. So a very high tessellation, high density tessellation of the higher order facets. But if the model grows pretty big, then we switch to just using linear elements, or maybe just 2 or 3 triangles per facet.
All right, thanks, Steve. That was a great talk. You have virtual applause coming in from Discord, so thank you very much.
Sure, you bet.
All right, so I'd like to thank all of our speakers once again for all of the great talks. The panel discussion was very interesting. Thanks for attending VisInPractice. It was a great year, even though it was virtual. I hope to see you next year in person. Thank you.
Text alignment is a fundamental technique in many text-related domains. In this survey, we cover 3 text alignment scenarios: collation task, text reuse detection, and translation alignment. On the basis of those scenarios, we reviewed the existing text alignment visualization approaches and discussed their advantages and drawbacks. We finally derived design implications and shed light on related future challenges.
ConfusionFlow is an interactive visualization for the performance analysis of classification models. It introduces a novel temporal adaptation of the confusion matrix, which lets model developers compare the learning behavior of multiple classifiers over time. Find out more about ConfusionFlow in our talk or in our TVCG paper.
Hands-on cybersecurity training represents a domain where visual analytics can significantly improve the impact of teaching process. We describe this new application domain and introduce a conceptual model that can serve as a framework for the development of analytical visualizations. Unified training lifecycle will be discussed from the perspective of different user roles.
We have seen many visualizations for tree data structures, but when showing changes in trees over time, some focus on displaying hierarchy, while others highlight their changes. With split streams, we combine two approaches and their individual benefits. We evaluated our method in a user study and consider it a general-purpose method for the visualization of dynamic hierarchies. Our JavaScript library is openly available.
Attention mechanisms have greatly improved the performance of many language models, yet with great power comes increased complexity. In this work, we present attention flows, a visualization that lets users interpret the language model's decisions and gain insights into the underlying self-attention mechanism. We also support model comparison that helps to fill the gaps between models in different training stages.
We propose an interactive ensemble analysis framework that provides flexible interactive exploration of the ensemble data. Time series characteristics of data can be obtained by fast browsing time steps. The region stability heat map view shows the stability of the selected region and provides region adjustment by directly clicking.
Deep neural networks are vulnerable to maliciously generated adversarial examples. This brings high risk in applying these networks to safety-critical applications. We developed a visual analytics approach to explain the root cause of such wrong predictions. Our contribution contains a constrained datapath extraction method, a river-based visualization, and a contribution analysis method.
In our current work, we visually compare two cohorts of segmented and classified tissue images, originated from single-cell omics data. To that end, we developed data-driven and interactive workflow, implemented in a multi-linked view system. Finally, we show the effectiveness of our workflow through specific case studies.
Tableau helps you see the stories in your data. It's designed to help you be smarter, so you can make better decisions faster. Connect to the data you care about. Sort, highlight, drill down, or filter your data in seconds. Add calculations to extend your data. With Tableau, you can keep on asking questions in the data until you discover the root cause. Tableau, answer questions at the speed of thought.
Morse complexes have shown a great utility in understanding the topology of complex scientific datasets. The noise in the scalar field, however, can significantly distort the Morse complex topology. Hence, we study the extraction of Morse complexes for ensembles of noisy scalar fields through their uncertainty visualization using our proposed statistical summary maps.
Program developers spend significant time on optimizing and tuning applications. But working with binary code to understand what compiler optimizations were applied can be challenging. We present our visual analytics system, CCNAV, designed to identify and assess compiler optimizations in binary code. Check out our paper to learn more.
KaraxS is an authoring system which enables rapid creation of zooming-based scatter plot-like visualizations in just tens of lines of JSON. We contribute a declarative grammar for such visualizations, which captures a large design space. By working with multi-node database spatial indexes, KaraxS enables interactive browsing of billions of data points. Come listen to our talk to learn more.
In this talk, we introduce CallFlow, an interactive visual analysis tool that provides a high-level overview of performance profiles from large-scale scientific applications. CallFlow visualizes the flow of resource of a program execution as a Sankey diagram to detect performance bottlenecks in parallel codes. CallFlow simplifies these calling context trees based on semantic information by constructing supergraphs.
Progressive visual analytics provides intermediate lizards in the middle of computation to address a long computational delay. However, such intermediate lizards are uncertain, so findings from these lizards can turn out to be incorrect for the entire data. In progressive visual analytics with safeguards, we suggest to risk the uncertainty, but we allow humans to leave safeguards every time they risk the uncertainty.
Imagine you have tons of text data to analyze. And you want to get an overview of your data, but traditional topping modeling techniques such as LDA are not working for you. Then, why don't you try Architext? We introduced a scalable and flexible way to interactively build hierarchical topics.
Digital humanities present great opportunities for testing new visualization approaches and evaluation techniques. However, and given the diffuse character and novelty of the field, it may also be intimidating for novel and senior researchers willing to get started in the discipline. In this paper, we propose a data-driven analysis of visualization for the digital humanities to identify key themes, authors, and relevant publications. So if you want to know more, please read our paper.
As data is changing, our understanding of data should be updated correspondingly. Based on machine learning approaches, we formulate a drift-level index to monitor the evolution of multi-source data, which allows users to capture and reason significant changes from time series data. The proposed visual analytics system is called Concept Explorer. More details can be found in our talk.
In our paper, we address the problem of navigating complex multiscale and dense environments, such as these molecular models. We present a technique for browsing a model by clicking on textual labels, which we call hyperlabels. This allows the user to intuitively navigate the hierarchical organization of the model. For more details, read our paper or watch the talk.
Entities and their changing relationships can be modeled more precisely as temporal hypergraphs, but hypergraph models are difficult to explore and refine. By leveraging domain-specific geometric deep learning models and a new multi-level hypergraph visualization, our technique allows for the direct integration of domain knowledge into the machine learning process. The multivariate hypergraph model structure can be analyzed on different abstraction levels. Simultaneously, experts can integrate their domain knowledge on the fly, and then explore the refined machine learning model.
Scatter plots and parallel coordinates are widely used for visualizing multivariate discrete data. However, for multivariate continuous data, the continuous scatter plots and continuous parallel coordinates are more suitable. In this work, we extend them to uncertain data. Additionally, we also extend fibers to uncertain data. If you are interested, come to our presentation.
Suppose you want to see the overcoming structure of a new project repository. Using Git client tools, GitHub Network, or Git log command is quite burdensome to get overview and navigate data. We presented Githru as an interactive visual analytics system for the Git metadata to help users explore and understand the context of development history.
We present Lyra 2, a system for designing interactive visualizations by demonstration. To design interactions in Lyra 2, users directly manipulate the visualization canvas. The system interprets demonstrations using heuristics and suggests possible interaction designs. We find that Lyra 2 enables rapid prototyping of an expressive range of interaction designs. We're excited to share this work with you.
If machine learning were like education, we would like to test what concepts our student, the model, has learned. Has it learned the concept of object rotation? Does additional text help with object recognition? We need a methodology and platform for conducting such tests. In this paper, we present a novel visual analytics tool that enables hypothesis-based evaluation of machine learned models.
We demonstrate that people can use the spatial cues available in virtual reality to help them effectively remember and recall scholarly articles. We used a virtual coffee shop and asked participants to remember 4 abstracts from scientific publications, and we termed this method a virtual reality memory palace variant.
We present VisInPractice, a tool for visualizing hypergraphs where edges can connect any number of nodes. VisInPractice is the first technique to display hyperedges with no overlap or crossing. Vertices are represented as rows, time periods flow from left to right, and groups can be shown on the left. We designed the layout for readability. VisInPractice shines with mid-size hypergraphs and it allows detailed analysis. We tested VisInPractice successfully using publication datasets and data from historical documents.
We present TransVis, a design study that is proposed to analyze and integrate close and distant reading of multiple translations. TransVis presents the overview of the collection to capture global patterns that is facilitated by the ADM-VIV matrix. TransVis integrates a detailed view to explore interesting path of alignments. We also propose the TLC view to examine and explore the terms of the user selected path to justify and reason the AD analysis.
Serine graphs are variants of stack graphs with curve-based line, and the main factor affecting its readability is the sign allurance through a perceptual inconsistency of the orthogonal and vertical direction. Aiming at reducing its impacts, we revisited the baseline formulation and proposed the concept of composition to help the serine graph layout optimization. The result shows that our algorithms perform better than the others.
How many clusters can you see in these images? With different visual encodings in scatter plots, our perception of cluster count changes. We developed models that consider how visual density influences cluster perception. Further, we demonstrate using a threshold plot to optimize the saliency of clusters.
Taylor-Coyer flows is a turbulent fluid motion created in the gap between the 2 rotating cylinders. The traditional reason-based methods are only able to capture the small-scale coherent structure. The detection of the large structure depends on the 2D cross-section, which is not the true configuration. We adapt the feature-level set methods and the density estimation to combine multiple attributes and use them as the filter to separate large and small-scale structure.
We asked participants to recreate bar graphs. When the bar was alone, we saw a different pattern of error than when the bar was presented with context in a stacked bar graph. We find that absolute error increased for higher bars when they were presented alone. We also found that there was a bias in the responses such that they were repulsed from an implicit 50% mark. We found a magic number, 10%, to predict error. Participants are usually within 10% of the height of the bar.
Tableau helps you see the stories in your data. It's designed to help you be smarter, so you can make better decisions faster. Connect to the data you care about. Sort, highlight, drill down, or filter your data in seconds. Add calculations to extend your data. With Tableau, you can keep on asking questions in the data until you discover the root cause. Tableau. Answer questions at the speed of thought.
Position is believed to be the most precise encoding channel, but our perception and memory of it can still be biased. Passwork has shown that the vertical position of bars in a bar chart is recalled in a biased manner, illustrating both an underestimation and overestimation bias of position. Here we find that the aspect ratios of the bar marks can cause this position bias, specifically resulting in overestimation for bars with wide ratios and underestimation for bars with tall ratios.
The classifier trained on the historical data may fail to classify the incoming data when the data distribution is changing. This phenomenon is called concept drift. To handle the concept drift, we developed Drift VIS, a visual analytics system to help the expert understand when, where, and why the drift happens, and update the model to improve performance.
The success of a convolutional neural network has been attracting many students and practitioners to learn the exciting technology. However, for beginners, the CNN model is not easy to understand. We introduced the Explainer, an interactive visualization tool to help beginners more easily learn about convolutional neural network. Using the Explainer, users can progressively explore the CNN model with real-life images in their browser, getting a comprehensive understanding of both high-level model structure and low-level underlying mathematical operations.
Out-of-distribution samples refer to the test samples not well covered by the training data, like these black cats. They are misclassified with high confidence due to their black bodies. To explain why these samples are out of distribution, we developed OOD Analyzer, a visual analytics tool which provides an ensemble detection method and a grid-based visualization to detect and analyze out-of-distribution samples.
Spot SDC is a visualization system that helps researchers understand the resiliency of HPC computation kernels to silent error corruptions. It gives users multiple perspectives of details with different granularity about the impact of SDC on an application's output. It also provides novel insight into how silent error propagates through a program's execution.
Several recent studies advocated the use of non-parametric density models for the improved characterization of data uncertainty. The non-parametric models, however, present the challenges such as increased memory and computational requirements. In this work, we propose an efficient non-parametric framework for volume rendering in the context of uncertain data and show their effectiveness in classifications via comparisons with the other statistical models.
Asteroids have always been and will always be a threat to life on Earth. Most likely, an asteroid will hit deep water, but what properties will actually lead to a threat like a tsunami? Ensemble simulations are a common tool to understand the effect of different parameters like the size of the asteroid. We propose an interactive visual approach to help analyzing deepwater asteroid impact simulation ensembles.
As commonly known, dimensionality reduction techniques and their interpretations are complex, biased, and uncertain. In drug design, this highly complicates the search for similar and new chemical compounds. To overcome this issue, Canva brings comparison of different molecular descriptors and properties into one tool, featuring planar, 3D, and table views for evaluating the trustworthiness of high-dimensional data projection.
When developing a deep learning model, people typically struggle to optimize hyperparameters that affecting the model performance. To relieve the pain of this manual trial process, AutoML has been developed as a solution. However, to maximize the potential of the AutoML, people should identify a good AutoML setting for their task. HyperTender helps to maximize the potential by visualizing AutoML algorithm behavior and guiding it.